Nitzan. Welcome to MedTech Unboxed.
Thank you very much. It's great to be with you guys.
Excellent. It's good to have you. I know we've been trying to set this up for a while and we finally have you. So I'm looking forward to our conversation and learning about the one and done LuSeed DOME implant.
I hope to give you guys as much knowledge and perspective as I can and to give some useful information to your listeners.
Okay, thank you. Let's get started and just kind of give us a background on LuSeed, and then we can talk about the unmet needs and the treatment of aneurysms. I know that 1 in 20 people are affected by aneurysms. You have a great video that talks about that, and we can go through the tech platform of the dome and maybe some of the economics involved. It's always, when you're a startup delivering new technology, economics are a very important part of the kind of the trifecta, right?
Always.
Yes.
Yeah. Eventually we build the business. It's inevitable. Yeah.
Yes. And then we can talk about any preclinical milestones in your pathway to, I don't know, maybe 510K. We can talk about that as well.
Okay. PMA, but thank you. Yeah.
Okay. Yeah. All right. Yeah. Tell us about LuSeed.
Okay. So we started about, you mentioned aneurysm, you mentioned a little bit of statistics. So all of this is correct. It's one of the more prevalent pathologies in the space, in the neurovascular space. And it's estimated that 1 out of 20 patients of the people harbors an aneurysm. And this resonates in hundreds of thousands of deaths due to ruptured aneurysms each year in the US alone, globally. If I'm not mistaken, this is the second biggest cause of permanent damage, morbidity or mortality after ischemic stroke. So it's a big thing. It's an event. And when we look at ways to treat aneurysms in the brain, so I'll walk you through the process over the years. Up until 30 years ago or so, these were treated by open surgery, clipping. And the endovascular or minimal invasive technology was first introduced by coiling, long, soft metal platinum wires that were put into the aneurysm in order to stop or prevent bleeding. And they were used along with stents to support the aneurysm that were wider in their opening. And these caused complications and the necessity of a very, very high physician skill to do it. It's still considered nowadays, by the way, the standard treatment. But because it's so complicated, new modalities were introduced. And these were called flow diverters, that are like stents that are implanted near the aneurysm intended to direct the flow away from it. But because these devices are usually implanted in the vasculature, the complication rates and limitations are still there. So a completely new way of thinking was introduced into the aneurysm treatment world. And these are called intrasaccular flow diverters. What does that mean, this complicated word? It means that there is a device, usually nitinol based, soft device, which is implanted into the aneurysm easily and quickly, and it's intended to prevent the bleeding. Or stop the bleeding. So it's kind of bring the simplicity aspect of flow diverters, but place it inside the aneurysm, like coiling, and prevent all the issues of having the devices in the vasculature, in the brain.
Yeah.
However, this is a new technology. I mean, there is one commercially available device in the United States, a couple of ones in Europe, a couple of ones in pivotal studies. So it's a very new technology. And as such, it suffers from the limitations of new technology. The first one and most obvious one is the long-term efficacy rates. And when we say long-term efficacy in the world of aneurysms, it means what is the proportion of aneurysms that are completely occluded, meaning zero chance that they will return. And with these devices, it's 50 to 60%. This is the official data, which is far, far, far from being gold standard. This is not what we need. We need something that works almost every time. Moreover, they are not fit for every case. Aneurysms roughly divide into bifurcation and sidewall, depend on the location of the aneurysm and what's called the morphology. So these are more suitable for cases with easy access. because of their geometry. So they lose almost half of the cases, so almost half of potential patients because of the structure. Moreover, they come normally with double-digit number of sizes. And imagine what happens if you're a physician and you need to select the right size, the one device that would be right from 40 availabilities to treat an 8 millimeter aneurysm or so. And if you choose the wrong one, the treatment will not be optimal. And this is in a resolution which is not like my and your screen. This is X-ray resolution shot in live in 2D. So it's not ideal. And this happens often in 2 a.m. with a bleeding patient under pressure. So all of these vectors, all of these inputs have led us to develop the LuSeed DOME. And this is a device that was developed intentionally to treat both types of aneurysms with similar ease of use of approach and deployment because of the design of the device, which is unique, has unique characteristics. And the first one, that it has no element that is dangling out of the aneurysm, meaning you have nothing out of the aneurysm, you have no protrusion to the parent artery, which makes the case selection much easier. We have short procedural time, 15 minutes or so in general. We come in 7 sizes so far, 1-digit number of sizes, which makes it easy. And with over 20 patients treated so far in our early feasibility program in Europe, we have over 90% of long-term occlusion, which is unprecedented in the space. So this gives us a confidence to expand our study and starting preparing for a pivotal program in the EU and the US. And I'll go a little bit backwards. So I'm a biomedical engineer in training, father of 2, live in Israel, in Tel Aviv. My dear partner, Amir Arthur, I'm the co-founder and CEO. Amir Arthur, my partner, is the co-founder and CEO of the company. He handles all the R&D, IP creation, strategic product development, and we together have a team in Israel, and now the company is further expanding and becoming multinational as we are moving closer and closer to our pivotal program. And this is a fully dedicated medical device company with R&D teams, QA, RA, clinical affairs, operations, logistics, and everything that has to do with it. So this is our story in a nutshell and what we came to solve and how we're going to do it utilizing our product.
Okay. Excellent. Thank you for that very comprehensive background. Some of the challenges you mentioned 50% with, I think, the closure device and maybe 90% with the dome. That's compelling.
It is compelling, and it's our job to keep producing this data in the expanded clinical studies. But yeah, so far this is the outcome.
Yeah. One of the things I read is not only the ease of use, but there's a potential compatibility with robotics. Is that something you can touch on?
Yeah, I can. Actually, I'll give an anecdote. Professor Vitor Pereira from St. Michael's in Toronto is the end-of-life research and innovation in the hospital, and he's also the head of LuSeed Scientific Advisory Board. And he's very close to robotics. He actually performed, to my best knowledge, the first thrombectomy procedure, which is done by an off-site physician performing the procedure. And we have designed LuSeed DOME. We understand that robotics and specifically off-site long-range robotics are one of the pillars of the future of the industry. Therefore, we've designed LuSeed DOME to be compatible with robotic systems. And the reason that makes it compatible with these systems is exactly what makes it easy to use. Because of the device is does not have any elements that obstruct the implantation and obstruct the arteries around it. And the easy maneuverability of the device and the predictability of the implantation. In fact, the predictability of the implantation is the one aspect that was repeatedly emphasized in conferences where LuSeed device was presented. For example, the last ABC Win seminar in Val d'Isère, which is one of the biggest conferences in the industry. So the physicians, Professor Sirakov, which had talked about his early clinical experience with LuSeed DOME, was explicit about the device predictability that, in his words, almost no matter where you place the microcatheter, the device would utilize roughly the same finalized position, which means it's very easy to determine if they like the device placement or not. And it's very makes it intuitive for a robotic system because you guide, when you use a robot system, you guide the device with your controller, which is outside the cath lab. And when you hit the deployment, you know that the selection is easy. Either you like the implantation or you change the device. This is the roughly the decision-making tree that you have. Unlike other devices that maybe you say, okay, maybe I need to go from the left, maybe I need to go from the right, maybe I need to create a micro angle in this and that, which is twice as difficult with robotic systems when you don't have the hands-on control. So therefore, we strongly believe that integrating LuSeed with robotic system would be natural for us. And it's designed to be that way. So thank you for raising the robotic system aspect.
Absolutely. If it's okay with you, there's a nice video animation that you have on YouTube, but I like to kind of have you walk us through it.
Yeah.
Okay. Let's see if I can. Okay. All right. I'll hit play here and let you walk us through this. If that would be okay.
Go for it.
Okay.
So yeah, here it's a simplification of the pathophysiology of aneurysm. It shows how slowly over time the aneurysm emerges because of instability of the vascular wall. And this is the LuSeed device illustration emerging out of the catheter. You see the two layers of the device which are not connected at the end. This is a unique feature of LuSeed. Coming out slowly and controllably out of the device. And when the microcatheter is guided through the arterial system, soon you will see it slowly coming close to the aneurysm space. It's the microcatheter distal end is located in the middle of the aneurysm, and soon you will see the device slowly being unsheathed into the aneurysm while the catheter is being retracted and capturing most of its volume, creating an inner stability which allows the device to remain adjacent or opposed to the vascular wall over time, even in highly hemodynamic environment regarding pressure and flow regimes. And as you can see, the blood flows freely around the device because you have nothing that's impeding its flow, and therefore what you saw, the simplification of the neointimal formation forming over the device, is what actually happens. And the combination of the flexibility of implantation and ease of use, having nothing outside of the aneurysm apart from a continuous, smooth dense nitinol mesh layer, and the device ability, because of this geometry, to be extremely stable over time. This is what we believe would give us the ability to really compete the gold standard, to provide the triangle of critical value drivers that we believe are needed, which is long-term performance better than any other device, ease of use, and the ability to treat various cases. So this is what we aim to bring to the table, and so far the team is doing a very good job in making it happen. And it's our responsibility to continue generating this data in large numbers on the pivotal program.
Okay, excellent. Well, thanks for walking us through that.
My pleasure.
A couple things that you mentioned. You mentioned that it's a dual structure scaffold, we'll say.
Yep.
With high density and nitinol, and it's delivered. That nitinol is just a beautiful material, a shape memory.
Yes, it is.
Super elastic. To unsheath, it kind of does a wonderful job. And there's no other material that can do that.
That's correct.
Yep. Yeah. And then some of the economic benefits, that's always a big deal, right? The ease of use and maybe shorter procedure times and some other things. Can you kind of walk us through those again real quick?
Sure. So when we talk about ease of use and procedural time, it resonates in two potential vectors of value generation. The first one is very strict, very financial, very spreadsheet-like. Quicker procedure time means more procedures per day, means more operational success, less financial burden on the hospital, less patient queue. And from a strictly financial standpoint, it's more insurance payoffs that are rolling through the system. From the operator side, it's the ability to cure more patients, because there are 24 hours a day. It will not change as long as it's the Earth will have its velocity unchanged. This will remain the same. So therefore, having a faster procedure is of interest in the extremely tight neurovascular space, where you don't have a lot of centers, you don't have a lot of big centers, and the patient load is immense, and any mean to reduce the patient load by becoming more efficient is a blessing, both from the clinical and the operational standpoint. And if we go a little bit from the system to the patient itself, their interest is to be as less time in the cath lab as possible, because the longer you are in the procedure, the longer one is exposed to the risk of thromboembolic events, because he has a bunch of metal in his arteries while doing the procedure. With all respect to antiplatelets that are being administered, there is always a risk. So less time with metal in the arteries, the better. Less time under X-ray, always the better. And less time under anesthesia, especially for patients with a little bit of kidney disease or whatever, is better. And this also resonates in shorter recovery times and everything that has to do with it from the get-go. So it's everyone's interest, both physician, patient, hospital, and financial spreadsheets, quote, to have a short and efficient procedure. And this is one of the main reasons where we, in our design, one of the constant variables was to keep one-digit number of sizes, always. Once you cross the two-digit, it becomes exponentially more difficult to change the device. And changing device, we believe, is the number one factor for a longer procedure time, because you just double the procedure time. If you deliver a device and you say, oops, it's too small, it's too big, you change the device. And we know that there are implementations with commercially available devices that they do 3 to 4 changes of device in one procedure. For us, it's a waste of time. It's something to be mitigated, and to be mitigated now. So this is the way we look at it.
Okay. Yeah. Thanks for walking through that.
My pleasure.
Yeah. Can we wrap things up with any updates from a preclinical side?
So from the preclinical side, first of all, we are a clinical stage company.
Oh, okay.
So we have treated more than 20 patients, and our long-term efficacy rates, which are so far compelling, are what's allowing us to get to the pivotal program. Of course, every patient, 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 ever aspect of the way was is following a highly meticulous and detailed preclinical program with biocompatibility, V&V, simulated use, and in vivo studies to ensure the product safety, both local and systemic, usability, feasibility, and of course, efficacy. And this was the gateway to even start treating the patients. So yeah, by all means, preclinical is immensely important.
Okay, absolutely.
Yep.
Yeah, and any updates on kind of the commercial funding or commercial strategy that you can share to wrap things up?
Definitely. LuSeed. So I cannot, I'm not in the liberty of discussing all the cracks of our fundraising, but we are fortunate to be backed up by high-tier medical device VCs and some companies from the industry, which are product with proficiency in products such as ours, and also institutionals and private equity, high net worth individuals and private equity firms. So it's a diverse knowledge of investors, and our board comprises both financial representatives and industry professionals, which brings years of experience, including commercial success in building a business in the neurovascular space, and both in building the commercial business and with creating high-value M&As for the shareholders. So this is something that right now we are expanding in order to even multiply the network of companies that are a part and partner with the company in order to ensure its success in the pivotal program and later in the commercial space in US, in EU, and any other potentially strategic territory.
Excellent. Looks like, the key point I think you said there, I hear a lot, is sometimes the boards of early stage startups don't have kind of the commercial aspect on their board. So you have that covered. So it looks like it's...
What? Can I share with you something? One of our board members, which I have some white hair, but you don't see because the Zoom is very flattering. So he has earned his white hair, and he says that companies are not getting sold; they are getting bought. So from the get-go, our goal is to build a company to be a successful business, and only that, the option of being bought would be on the table, and we would have the luxury of selecting the best alternative for the company.
Nitzan, thank you so much for joining. This has been a very interesting conversation. I'm really looking
Steve, it's
forward
my
to it.
pleasure. It's better late than never, so they say, and I'm really glad that we had a conversation. I hope that I provided value for your listeners with interesting knowledge and understanding about the space and about what we came to improve as our humble part in the evolution of neurovascular devices. And thank you. I appreciate it.
All right. You're welcome. Thanks again.